So antibiotics, as usual, comes from Greek, the root words, not antibiotics, not antibiotics, the things. Those are a little bit different. Anyway, antibiotics are chemical agents that prevent the growth of living organisms by targeting some aspect of cellular machinery. So under this definition, antibiotics include antibacterial agents as well as antifungal, pesticides, chemotherapeutic agents. They're all antibiotic. Anything that prevents life. So antiviral medication is not antibiotic because viruses, as we learned in Unit 2, are not life. Viruses are not alive. Viruses never lived in the first place because we have our 5 defining features of life. Our cell is our basic unit of life, and viruses are not cells. So antiviral medication is its own thing. Antibiotics are for cells, against cells. So we have 2 flavors of antibiotics, and then we have 2 spectra. There's bacteriostatic, in which case the bacteria are prevented from growing and multiplying, and the host immune system or the environmental factors are relied upon to reduce the numbers of bacteria. And then we have bactericidal, which kill bacteria through direct action. Then you have the spectrum. The spectrum is the range across which an antibiotic is effective, the range of species. If a bacterium is effective against many different species, it is a wide spectrum antibiotic. If it is a narrow range, then it is only effective against a few very specific particular species. So a wide spectrum bactericidal antibiotic is an antibiotic that kills many different species of bacteria directly. Through direct action, it kills them. It reduces their numbers. A narrow spectrum bacteriostatic antibiotic only works on a very small number of bacterial species and prevents their growth and multiplication. So these are 2 adjectives that you would use to describe antibiotics. I'd like to remind you of 2 things, though, from Unit 1 and Unit 2. Enzyme inhibition from Unit 1. First, enzymes. Enzymes are globular proteins that reduce activation energy of a chemical reaction. Enzymes have an active site that allows a substrate to bind and lowers that activation energy for that chemical process, allowing the reaction to proceed. Without an enzyme, normal biological processes would not proceed because there is not enough ambient energy as measurable as heat or measurable as temperature to move that reaction forward by normal chemical means. A catalyst is required to move that chemical reaction forward. So enzymes are required to move chemical reactions forward in biological systems. Enzyme inhibitors. The word inhibitor, the word inhibit, means that it will prevent, it will stop the function of an enzyme. An enzyme inhibitor is something that prevents the function of an enzyme. An enzyme is unable to perform this lowering of activation energy of that chemical process. And you have many different means of enzyme inhibitors. Depends on where that inhibitor works on the enzyme. If it works by binding to the active site instead of the substrate, then it is a competitive inhibitor. If it works by binding elsewhere and does not affect the binding of the substrate, it is a non-competitive inhibitor. The substrate can bind just fine. the inhibitor prevents the activation energy from being lowered, prevents the reaction from proceeding. Allosteric inhibition is an inhibitor will bind somewhere else, not the active site, but it will change the 3D conformation of the enzyme, preventing the binding of the substrate or the release of the substrate. It prevents the reaction from proceeding by changing the active site, preventing or changing enzyme specificity, enzyme affinity. Bacterial cell walls. The cell wall of a bacterium is different from the cell wall of a plant. The cell wall of a plant is composed of cellulose, repeating units of beta-glucose in alternating up-down, up-down, up-down units, flipping the molecule 180 degrees each time. And then they're arranged in chains that are stacked together into microfibrils, and then into fibrils, and then into fibers, and then you stack the fibers into that wall. Bacterial cell walls, or prokaryotic cell walls, are composed of something else. They're composed of peptidoglycan, which is a composite of a polypeptide and a carbohydrate. It is not composed of cellulose. So many prokaryotes have cell walls. They'll have, if they have a cell wall, they'll have an inner membrane and a cell wall, and they may or may not have an additional outer membrane as well. And they may or may not have an additional slime layer or a capsule. So these peptidoglycan cell walls, they need to be cross-linked. The peptides themselves of this cell wall needs to be cross-linked in order for it to retain its consistency. And the cross-linking of those peptides is performed by an enzyme called transpeptidase. This enzyme called transpeptidase becomes important. Let's talk about penicillin. Penicillin is derived from the Penicillium genus of fungus. It is an antibacterial. Its properties were first discovered by accident by Alexander Fleming in 1928. He freely admits this. Well, admitted. I think he's dead now. Pretty sure he's dead now, because otherwise he'd be well over 100 years old. And that's rare. Yes. So his plates got contaminated by fungus, and before he got around to throwing them away, he noticed that wherever the mold was growing, there was an area around them where the bacterium would not grow. This area of no growth, he studied and concluded that there was some chemical agent killing the bacteria in this region of no growth. Since the discovery of penicillin, which you should actually spell out, penicillin. Don't abbreviate it PCN for the Cambridge exam. I did that for my own sake because it was easier to write the slide that way. Just write it penicillin, write it out. Write it out. So many different antibiotics have been derived from penicillin since then. So the core of the structure, the core of the chemical structure of penicillin is this beta-lactam ring. This 4-member ring with a nitrogen and a ketone group coming off of it. This is the beta-lactam ring. It is the core of this chemical structure. This is penicillin over here, and these are cephalosporins. Some individuals are allergic to penicillin, And in some cases, that allergy carries over to cephalosporin antibiotics as well. It doesn't always, because there are significant differences in the shape. This group over here is different, is considerably different in shape, so the reaction of the immune system is different in most cases. But sometimes that reaction is similar enough that an allergy can be produced. But this core structure allows these to be referred to as beta-lactam antibiotics. This chemical structure, this ring, the beta-lactam ring, makes them beta-lactam antibiotics. So let's talk about the mechanism of action of penicillin. Remembering for a moment that I specifically just went over the physical three-dimensional structure of penicillin. And the three-dimensional structure of a molecule is important when you're talking about enzymes. Penicillin works by irreversible, competitive inhibition of transpeptidase. Irreversible inhibitors create a dead-end complex. They form a permanent covalent bond with whatever it is they are inhibiting, making that enzyme no longer functional. Competitive. They are competing with the substrate of the enzyme they are inhibiting. They are competing with the substrate for the active site. And the only way penicillin can compete for the active site is if it is shaped similarly to the substrate of transpeptidase. Transpeptidase is a class of protein that has been labeled penicillin binding proteins. They are a bunch of proteins that are part of building and restructuring and resurfacing the bacterial cell wall. So transpeptidase is the one that you need to know for the Cambridge exam. So any transpeptidase enzyme that is made that comes into contact with penicillin for long enough will be permanently inactivated, permanently inhibited by a penicillin molecule. So long as penicillin remains in the system, any new transpeptidase will be inactivated. So when you cannot remodel the cell wall, the bacterial cell wall, a bacterium that attempts to grow and divide will have weak points in its wall. One of these weak points will eventually rupture, and the inner membrane of this bacterium will herniate through. It will herniate through, rupturing the bacterium, creating a spiroplast, which is a wall-less bacteria of a previously walled bacteria, and all the protection that wall formerly provided is gone. The cell wall served a purpose in this bacterium, and now that purpose is no more. That purpose was structural integrity, protection, and prevention of lysis from osmotic factors. Eventually, this spiroplast is going to burst because it is saltier inside that spiroplast than outside. Water will follow salt, and it will enter this and burst it. So this is a bactericidal antibiotic. But it only functions as a bactericidal antibiotic when the bacteria is growing. If it is not growing, there will be no herniation. There will be no remodeling of the cell wall. So the bacterium that are trying to grow will die. Die by direct action. But this is killing them. So it is bactericidal. It is murder. bacterial murder. So you will need to be able to do your own, you will need to be able to explain why antibiotics work on a bacteria and not on a human. You'll need to be able to explain why antibiotics do not work on viruses. The key here is to recognize and understand that the target of an antibiotic and its mode of action work in a very specific way. And if that target is present in a bacterium, it will work. If it is not present, it will not work. And targets that are present in bacterium and not in humans or in plants means that it will not work on humans or plants. So, if they give you a sample antibiotic that functions on rupturing lysosomes, it will not function on it will not function as an antibacterial agent. It will function against eukaryotes. Why? Because lysosomes are membrane-bound organelles and prokaryotes do not have membrane-bound organelles. It will kill eukaryotic organisms. An antibiotic that works by binding to the small ribosomal subunit of the 70S ribosome. Why would that work on a bacterium and not a human? Because bacterial ribosomes are 70S and eukaryotic ribosomes, humans are eukaryotes, are 80S. They do not have the same ribosomes. The antibiotic will not function on the ribosome of the eukaryote. It will only function on the small ribosomal subunit of the prokaryotic bacteria. And by the way, streptomycin, tetracycline, those function on the 30S, which is the small ribosomal subunit of bacteria. Erythromycin and clarithromycin function on the 60S, the large ribosomal subunit of the 70S. Oh no, the 50S, sorry, the 50S subunit, the large ribosomal subunit of the 70S ribosome of the prokaryotic bacilli, a prokaryotic bacteria. They won't work on humans because humans have a 80S ribosome. Their large ribosomal subunit is 60S, not 50. The 50S is what it acts upon. I remember it as 50C to 30th Street, but long story. So, why won't antibiotics work on viruses? Because viruses aren't cells. Cells are living things. Viruses are not cells. Viruses are not living things. Antibiotics are against living things. Viruses don't die from antibiotics because viruses never lived. So antibiotics work by targeting a specific cellular machinery. Viruses do not have their own cellular machinery. Viruses hijack the cellular machinery of their hosts. So unless you are targeting the host to kill the host, antibiotics won't work. And you don't want to kill the host to treat the virus. Otherwise, you might as well just shoot him in the head. Don't do that. That is not an effective way to treat a virus. That is a bad way to treat like anything. Don't do that. That's a bad thing. That's a bad thing all around. Okay, so here's some other ways that antibiotics can be tested. compare the efficacy of different types of antibiotics. This is a masked ring. This is a group of antibiotic soaked pieces of paper. And you test the efficacy of different antibiotics by measuring the distance from the piece of paper that has cleared the bacteria. That there is no growth. That's how effective the antibiotic is. So the most effective antibiotic is the one that has shown the most clearance. So if you're asked a question like, which antibiotic should be used to treat the patient? And you're given one that has 4 dots and one of them has cleared like a large space around it and the others have like no clearance, a little more clearance. Take the one with the biggest dot. Take the one with the biggest dot. That's actually a Unit 1 question. That's a Paper 1 question. Take the one with the biggest dot. That's the one that's the most effective against the bacteria. The one with the most clearance is the most effective, the best murderer of bacteria. Now, if you're asked to compare the efficacy or explain why one works better than the other, it's the same question as before. What's the target? Is it present in one versus the other? If one has no clearance and the other has large amounts of clearance, is the bacterium gram-negative or gram-positive, and what is the target of the antibiotic? If the antibiotic is the cell wall, targets the cell wall in one, and the other one targets the ribosome, and the plate of bacterium is a bacteria that does not have a cell wall, then if the bacterium does not have a cell wall, it's a gram-negative bacterium, then the antibiotic that targets the cell wall will have no clearance. It's not going to work because it's targeting something that isn't a target. It has no target to target. It won't work. But the one that the ribosomes, the one that targets the ribosomes, every cell has ribosomes. Otherwise, it wouldn't be able to make anything. It will not if it doesn't have ribosomes, it's not metabolically active. If it's metabolically active, you can kill it by targeting the ribosomes. It's one of the reasons why doxycycline can kill almost anything. All right. Now, remember that there are also questions about the diffusion, rate of diffusion by molecular weight. The smaller a molecule, the lower the molecular weight, the faster its diffusion will be. So if they are all the same mechanism of action, and there is no resistance mentioned in the question stem, then the one that spreads the farthest is the smallest molecule. They're really asking you a diffusion question. Compare the efficacy of different concentrations of antibiotics. If they show you a plate that has different concentrations of antibiotic soaked paper, the one with the largest concentration has the most clearance. The one with the lowest concentration has the least clearance. That's almost always going to be the case. And basically you would plot this on a graph. They sometimes might be able to ask you to determine from a graph of the data what the concentration would be that would give you this amount of clearance. That would be a graph reading question. So be prepared for things like that. Those are very basic things. So let's revisit penicillin for a moment. It works by irreversible competitive inhibition of enzymes. How would bacteria get around the problem posed by penicillin? Don't think of it as an issue of change everything about the bacterium. The bacterium has been getting along just fine before penicillin got introduced. The bacterium was doing okay. Then penicillin comes along and starts mucking up the works. Penicillin is the problem. Penicillin is the poison here. You get rid of the poison, there's no problem anymore. So that's the bacteria's train of, well, not thought, because it's a single-celled organism with no organelles. It's not really thinking. It's just, doing. You cut penicillin, or you pump penicillin out. This is by no means a complete list, but it's simplified for your purposes. And the bacterium can use to perform these actions enzymes and proteins. How do you make enzymes and proteins? You translate them from mRNA. You synthesize proteins by translation from mRNA. mRNA you synthesize by transcription from DNA. Mutations in the DNA can change the primary structure of proteins and could result in the formation of new enzymes. So, revisiting plasmids, Unit 2 stuff. Plasmids are small circular segments of DNA found in prokaryotes that can exist freely in the cytoplasm, away from the main nucleoid region of DNA. And while genes encoded on plasmids may or may not be useful to the organism, plasmids will often contain genes that confer antibiotic resistance. And that's a big problem for a number of reasons. Usually horizontal transmission, which we'll discuss in just another moment. Betalactamase is one such plasmid-mediated antibiotic resistance gene. It's formally known as penicillinase. Betalactamase is an enzyme that cuts the beta-lactam ring, which we discussed that structure of. We discussed that structure. It is at the core of penicillin and other beta-lactam antibiotics. If you cut the beta-lactam ring, you change its three-dimensional structure. And because penicillin acts as a competitive inhibitor, competing for the active site, which has three-dimensional specificity for a substrate, because it is highly specific for a three-dimensional shape. If you change the three-dimensional shape of penicillin, it's no longer competitive. You have hobbled it. You have made it no longer a contender. It can't compete for the active site of transpeptidase. It's no longer a danger. So it inactivates penicillin permanently. It may also inactivate other beta-lactam antibiotics. Some cephalosporins can be inactivated by beta-lactamase. Not all of them. Some of them are beta-lactamase resistant. But you can also add other drugs to penicillin, like sulbactam, for example. Sulbactam is a beta-lactamase inhibitor. So you inhibit the inhibitor. You inhibit the thing that is preventing penicillin. Beta-lactamase inhibitors, like sulbactam, prevent beta-lactamase from cutting penicillin, which allows penicillin to do its job. You're basically hiring a bodyguard for penicillin. So you give penicillin-type antibiotics together with something else to prevent them from being chopped up before they get to do their job. That's a way that you can combat resistance to antibiotics like penicillin by beta-lactamase, is you give something to stop the beta-lactamase. But let's talk about resistance some more. As we discussed when we were talking about TB briefly, and I promised I'd come back to it, now I'm making good on that promise. Resistance is when an organism is less susceptible to an antibiotic therapy. Antibiotics can act as a selection pressure. And when I say a selection pressure, I'm talking about artificial selection. When you kill off, when you have a series of bacteria, one of them is square, or two of them are square. I make an antibiotic So let's say that I have a group of bacteria. Some of them are circular and some of them are square. The circular ones are the regular ones, and the square ones are my antibiotic resistant ones. My antibiotic kills all the circular. My antibiotic one, the square ones are resistant to my antibiotic that kills circular bacteria. So I give the antibiotic, and it kills all of the circular bacterium, and maybe one of the square ones, because it's just resistant, it's not immune. But I wasn't expecting the square bacteria. Now the only one left is the square. And now it can divide into more squares, more squares, more squares, all the squares. And now all I have are square bacterium. And now I try and give my antibiotic again. And nothing happens. Because my same antibiotic that killed all the circular bacterium, there's no circular bacterium to kill. The square ones are resistant to my antibiotic. I might kill one or two because, hey, it's killing some of them. I might kill the old one, the weak, the petty, but they'll still be there. They're resistant, not immune. It's not as effective. It's not effective very much at all. That's selection pressure in a nutshell. I have selected for the squares by pressuring the circular bacterium out. I have put pressure on the circular bacterium until they are now gone. The squares are the only ones that remain. End of metaphor. I erased it because I was done with it, not because I added a new antibiotic. All right. So these are the two types of transmission of resistance for our purposes. There is vertical transmission and horizontal transmission. The one I just described was vertical transmission, where a bacterium divides from a progenitor bacterium, a parent, into its offspring. And when you remove all of the others, the ones that remain divide and grow. That's the selection pressure acting, and then vertical transmission takes you the rest of the way. Vertical transmission is when a gene is passed from one parent cell to its offspring cell, to its daughter cells. Horizontal transmission can occur in one of three ways, or multiple of three ways. You have bacterial transformation. And by the way, these are ways in which you can pass plasmids. That's why plasmids are problems for us. Transformation is when this bacterium here has been killed in a particularly gruesome death. Its DNA guts are splayed all over the battlefield. Another bacterium, previously naive in the ways of antibacterial resistance, is now searching the battlefield, picking it for useful remains. And it picks up a useful piece of still usable DNA that codes for a resistance gene. That is bacterial transformation. It has now consumed the fallen and transformed into a resistant bacterium, when it was previously non-resistant. It may not even be the same species. It is transformed by consuming the genes of the fallen. Bacterial transduction occurs when a bacteriophage virus infects a bacterium, and in the process of packaging its DNA, in packaging new DNA into its capsid, it accidentally packages some of the bacterial DNA into that capsid instead. And it delivers that phage to a new bacterium instead of the viral DNA. It's a case of incorrect receipt of package. This isn't my mail. Whose mail is this? I didn't order these books. But now the bacterium has a new gene that was given to it by this bacteriophage from another bacterium. That's bacterial transduction. Then there's bacterial conjugation. Recall, if you will, the specialized Fimbriae that was the pilus. The F. pilus being the most famous of these. Bacterial conjugation is when one bacterium with a pilus connects via a conjugal bridge to another bacterium and then starts copying its DNA over to the other bacterium. A plasmid may cross, parts of its nucleoid region may cross. Whatever crosses, it doesn't matter. Some parts of that bacterial DNA will transfer. And this new bacterium that has just been conjugated with has been the receiving end of the pilus, now has a new plasmid, a new set of genetic instructions with new genes, with new traits encoded on it. And those traits may encode for antibacterial resistance. So those are horizontal transmission. These bacterium that are receiving new genes did not inherit these traits from their parent cells. They received them from neighbors, victims, viruses, and the like. So some common mistakes. Both of these mistakes are going to cost you points, even if the context of what you say is correct in every other fashion. Stating that resistance to antibiotics develops in people is incorrect. It develops in bacteria. People are not resistant to antibiotics. That is incorrect. Bacteria are resistant. Do not confuse resistance with immunity. Bacteria are not immune. It just takes more work. Even if you are trying to pump out, if the mode of resistance is by pumping out penicillin away from the cell wall of the bacterium, you can give a higher dosage of penicillin to outpace that pump, and they will still die. It just requires a higher dosage to kill them. It's not immunity. It's just harder to kill them. Resistance. They resist dying, but they are not immune to death. No one is. So bacteria can develop antibiotic resistance. That is your correct statement. Do not make these mistakes. So, shameless plug. One of my other favorite channels, which if I remember, I'll put a link in the description, Healthcare Triage, has an episode on antibiotic resistance that pairs nicely with this information. You should go watch it.